Hi everyone, welcome to your first lecture on photosynthesis. First, we're going to talk about what are autotrophs and why they are important. Autotrophs are organisms that make their own food. They're also known as producers. Examples of autotrophs include plants, some algae, which can be protists, and some bacteria are also autotrophs. Heterotrophs have a different food source. You know that the word prefix hetero means different, and they need to eat. They depend on other organisms for their food. They're also called consumers. Examples like animals and some bacteria. We are heterotrophs. That's why we have a cafeteria in the school. There are two biological processes that are needed to create usable energy for organisms. The first one is photosynthesis, and that process converts sunlight into sugars, and only autotrophs can do that. So it can be done by some bacteria, plants, and algae, so some protists. And then cellular respiration. Do not confuse this with the ability to breathe in and out. Cellular respiration is done in the mitochondria in your cells, and it is taking that glucose, sugar, and turning it into usable energy for your cells. This is done by both autotrophs and heterotrophs, not just heterotrophs. ATP is that main energy storage molecule. So when we're talking about energy being created or used in the cell, typically we are talking about ATP. ATP stands for adenosine triphosphate, and you can see here that the molecule is made of an adenine, the ribose sugar, and then 3 phosphate groups. So that's why adenosine stands for the adenine, tri means 3, and then phosphate for the fact that there are 3 phosphates attached. ATP is the universal currency for energy, and that energy is stored in the bonds between the phosphate groups. And when a bond is broken, one of those phosphates breaks off, then energy is released. So what you get if you break one of those bonds off, you start with the ATP, or the adenosine triphosphate, and you convert that into ADP, or diphosphate, di meaning 2. And then when that extra phosphate is broken off, that's when you have your release of energy. ADP, the diphosphate, can absorb energy also and add another phosphate on to create that ATP again. So in your cells, this allows the cells to use just a little bit of energy at a time. When we're talking about photosynthesis in plants, it occurs in the organelle called the chloroplast, and I know that we have already spoken about cell organelles and the fact that photosynthesis happens in the chloroplast, and even seen some examples of chloroplasts in class. However, we need to know a little bit more about their internal structure to know how photosynthesis works. So the chloroplast is the structure in plant cells where photosynthesis occurs, and it has a few different parts. The first is a thylakoid membrane, which looks like stacks of pancakes or stacks of coins. The lumen is the space inside that thylakoid membrane. A grana is what we call the big stack, and then the stroma is the solution that is around the thylakoids. So next slide, we're going to go to a picture of it and see what this looks like. The thylakoid right here are these stacks of coin-shaped membranes. The granum is the entire membrane here. That's the granum. Inside of the granum is your lumen, and outside of those thylakoid membranes, that is your stroma. Within the chloroplast, there are pigments. Chlorophyll is the most common pigment within. It's the primary pigment within the chloroplast. And chlorophyll A and chlorophyll B both work to absorb different spectra of visible light. This is approximately the light that can be absorbed by chlorophyll. You can see that it absorbs a lot of the blues and quite a bit of the red, but it does not absorb the green. That is why when we look at plants, they look green. That color that is not being absorbed by the chlorophyll or the pigment molecules is being reflected back to our eyes, and that is what we see. So each pigment absorbs and reflects different wavelengths of light, and we see the reflected color, which is green. So if you could say, maybe what are plants' least favorite color, then that would be green. It's the one that they do not absorb that wavelength of light. So like I said before, chlorophyll is the primary pigment in photosynthesis, and it gives plants their green color because it reflects it. Carotenoids are accessory pigments that absorb light that chlorophyll cannot, and typically they are yellow, orange, and brown. So why would leaves turn yellow, orange, and brown in the fall? Because the production of chlorophyll is slowed down due to the fall having lower temperatures and less sunlight. So then you get to see those wonderful fall colors come out in the leaves because you don't have as much of the chlorophyll reflecting that green pigment. This is just a picture of that. The sun gives off white light, which is a mixture of all of the colors on the spectrum, and the one that is not absorbed is what we see, and for plants that is the color green. So the main equation for photosynthesis is 6 molecules of carbon dioxide plus 6 molecules of water plus sunlight gets converted into glucose and 6 molecules of oxygen. So your carbon dioxide, your water, and your sunlight are your reactants, and then your products are the glucose and oxygen. When scientists first started investigating photosynthesis in the 1600s, Van Helmont was a scientist that studied, starting with a kind of basic question. When a tiny seedling grows into a very tall tree, where does that mass of the tree come from? So he didn't know if it came from the soil, if it came from water, or it came from the air. So we're saying, how does this itty bitty little seed turn into this gigantic redwood tree, or, the big maple or oak that's in your yard? And where is it getting all of that weight, and where is it getting it from? So he did an experiment, and he measured plant mass and soil. Then he wanted to see if the soil would deplete or decrease in mass if the plant was getting it from the soil. And what he found over time was after 5 years, his seedling had grown into a tree, and the amount of soil that was there weighed the same. So the only thing he had really been adding was water. So he figured that most of the mass or the gain came from the mass of the water. However, then that is part of the equation. You do need water for your trees to grow. But he was missing a very important component, which is the carbon dioxide gas in the air. Here is a picture of his experiment. He had a little willow tree and 200 pounds of dirt. And so that is on day 1, a 5-pound willow sprig and 200 pounds of dirt. 5 years later, his willow tree had grown to almost 170 pounds, and he still had almost 200 pounds of dirt. So the mass of the soil did not decrease at all, but the tree had grown significantly. And since the only thing he had been adding for the tree was water, that is why he came to his conclusion that most of the gain of mass came from the addition of water. Another scientist that studied photosynthesis or plants in the 1700s was Priestley. And he covered a candle, a burning candle, with a jar, and he noticed that pretty quickly that candle would go out. Then he did another experiment. He put a little sprig of mint and the candle in a jar and noted that the flame of the candle would last a lot longer with the mint in there. That is because the mint sprig produced oxygen. Oxygen is a byproduct of photosynthesis. So Priestley discovered that plants produced a gas that the flame required to stay lit, which happens to be oxygen. This is his experiment right now of the jars he put over the candle and the flame, and then once he added the mint sprig, it lasted longer. And then Ingenhouse also studied in the 1700s, and he was able to show that the effect that Priestley had observed of the plant producing oxygen only occurred in the presence of light. He did the experiments with light and in the dark, and the production of that oxygen gas did not occur unless there was light. So he discovered that light was necessary for during the process of photosynthesis.